So let us start. Today's topic is the unit number 3, that is nervous system. So the first point we are going to discuss about it is about what is nervous system, introduction of that, how the communication takes place between neurons. Okay, then after that, in the next lecture we'll discuss about the brain structure, and then what is EEG, that is electroencephalograph, and what are the various techniques, that means the electrode positions for the measurement of EEG. So that is this unit number 3. So first, today we'll start with the beginning of nervous system. What do you mean by nervous system? The introduction of nervous system. Okay. Uh, so the human central nervous system, actually the basic building block of this nervous system is the neurons. These are the cells. So our complete nervous system, it is made up of cells called as neurons. So in our central nervous system there are more than 100 billion neurons, which are 50 to 100 times the number of glial cells. So these glial cells are again a different type of cells available in our nervous system. These glial cells do not take part in the communication. That means they do not generate any electrical signal. So that is the difference between this glial cell and the neurons. Okay. The role of glial cells is to protect these neurons. So we'll see some more points about these glial cells later. So the glial cells are 50 to 100 times, neurons are 50 to 100 times the number of glial cells. About 40% of human genes participating its formation. Now we also discuss sometimes about the muscles. So the function of muscle cells is for contraction, whereas the function of the neurons, it is integration and transmission of nerve impulse. So we have seen earlier in unit number 1, that is action potential. Okay, so under normal conditions the cells are in the polarized state. When they are depolarized, some action potential in the range of 25 millivolt it is generated, and then it is transmitted from one cell to the other cell. So in case of nervous system, those are neurons. So the special function of neurons is integration and transmission of nerve impulse. So the transmission of integrate the signal and transmit it from one point to the other point, that means from one neuron to the other neuron. Now along with endocrine, nervous system forms the major control system of body functions. Endocrine system is the system which is responsible for generating different types of hormones. So based on it is actually the feedback system. So based on the feedback, the different types of hormones are generated, and those are actually some chemical signals. So along with endocrine, nervous system forms the major control system for body functions. Okay. Now this diagram gives the overview of the nervous system. Okay. Nervous system, it is classified mainly into two categories: central nervous system and the peripheral nervous system. Okay. Now when we say nervous system, you must be aware that it starts from the bottom of our spinal cord and it terminates, that means the other end of the nervous system, it is at our brain. Okay, so we are discussing about the nervous system. Nervous system, it is classified into two categories: the central nervous system and peripheral nervous system. So as I said, the central nervous system, it starts at the bottom of the spinal cord, and through spinal cord the bunch of nerves, it extends through our neck and up to the brain, top of the head. That means in our head it is the brain. Okay, so the central nervous system, it is, as we discussed, it is composed of brain and the spinal cord, right? Then the other part of the nervous system, it is peripheral nervous system. So this peripheral nervous system, it is connected to the central nervous system. Okay, so the central nervous system, as it starts from the spinal cord and it extends up to the brain, so at various points some branches originate from the central nervous system to control the other parts of the body or the organs. Okay, so that part of the nervous system, it is called as peripheral nervous system. Now again, this peripheral nervous system, it has classified into two parts: the autonomous nervous system, automatic, or it is also called as autonomous nervous system, and somatic nervous system. Autonomous nervous system, that means this is actually the autonomous nervous system, it controls the various functions of the body automatically. That means without any extra effort from our side. That means, in other words, this connection gets disconnected. Okay, so peripheral nervous system, as we were discussing, it is classified into two categories: autonomous nervous system. So this nervous system, autonomous nervous system, means it works without making any effort, or actually the correct word for this, it is the subconscious mind. We call it as the subconscious mind. For example, when we are sleeping, so during sleep also our brain and various organs, they are working, heart is also beating, and all these things are controlled by through our brain even if we are sleeping. So that part of the nervous system, it is this autonomous nervous system. Okay, and somatic nervous system is the control signals what we pass on to the through the nervous system intentionally. Okay, so that is somatic, that means the conscious mind. Now again, this autonomous nervous system, there are two parts: sympathetic nervous system and parasympathetic nervous system. So this parasympathetic nervous system, it is actually it is working when we are relaxed, during the relaxation position. That means it will slow down our heartbeat. Okay, now sometimes example is, for example, there is a sudden fear, okay, or some you get some shock, suddenly some shock, right? So in that case, there will be sudden reaction from the body, heartbeat will increase and so on. So that happens because of the sympathetic nervous system. So both these nervous systems, both these parts, they are actually working together. So sympathetic nervous system, it will increase your heartbeat, and the parasympathetic nervous system, it will try to reduce the heartbeat at the steady level. So these are actually these two types of autonomous nervous system. So this is the, in broader sense, how our nervous system is classified. As far as our subject is concerned, our main intention of studying this nervous system is to, as an electronics engineer, is to learn the signals corresponding to brain called as EEG, electroencephalograph. So our more focus, it will be on brain. Okay, just to give you idea, I have told you these are the various different parts of nervous system. So our focus will be mostly on brain. That means EEG. What are the EEG signals? How they are produced? How they are measured? Okay. Now, as we discussed, the cells are made up of neurons, and there are many different types of neurons. Apart from neurons, there are some other types of cells in the brain called as glial cells. So, as I told you earlier, these glial cells are glial cells do not take part in communication. That means glial cells do not generate any electrical impulse. Okay, so its functionality, it is to protect the neurons. So brain is made up of very small discrete cells. There are about 100 billion neurons in the human brain. These neurons are like any other cell in the body with a membrane, nucleus, and specialized organelles. This is the typical structure of the neuron. This is the nucleus, that is the main part of the neuron, right? Then there are some nerve fibers originating from the main body of the neuron. So the cell body, it is called as soma. Body of the cell, it is called as soma, and the central part, it is called as nucleus, and from branches originate from that main body, those are called as dendrites. These are actually the nerve fibers. There is this bigger branch called as the axon. So this axon originates from one neuron, and it will connect with the other parts of the neuron, other neurons in the vicinity. So that is this axon. So it is actually the nerve fiber. Now at the end of this axon, there are smaller branches of these nerve fibers called as axon terminals, or the terminal branches. So this is the overall structure of neuron. The neurons have 4 important regions. Okay, so these dendrites, as we have discussed earlier, so these are the branching projections that collect information. So these dendrites, these are the branches of this neuron which collect information. These are various different types of neurons, what you see in this diagram, different diagrams. As we have discussed, the body of the neuron, it is called as soma. It contains the nucleus and it integrates the information. So this is soma, what you see in the diagram. So there is a nucleus, dendrites. Now this part of the axon, it is called as axon hillock. So this is the point actually where the electrical impulse or the action potential, it is going to originate. And this axon, it is coated by some myelin material called as myelin sheath, and there may be some gaps in between. Axons conduct neuron signals across long distance. So this is the axon, it is a nerve fiber starting from one neuron, and it may be connected to the nearby neuron. So the axon is actually conducting the signals from one neuron to the other neuron. Dendrites collect thousands of incoming signals, and soma integrates signals from dendrites, generates signal that will travel down the axon. So these dendrites, they sense the signals from various nearby sources. Then soma, it will integrate all these signals, and based on that it will generate some response in the form of electrical signal, and that electrical signal, it will travel away from this neuron through this axon, and it may go to the other cells. Axon terminals. So these are the small swellings that release signals to other neurons. Now the transmission of signals, it also takes place through some chemicals. So these are the chemical signals known as neurotransmitters that cross small gaps, and the small gaps are known as synapses. Okay. So it is estimated that there are about 500 trillion synapses in the adult brain. I will show you one small video clip to know what exactly is this synapse. So we'll go through the video clip, and then after that again we'll come back to this PPTs.
Welcome to 2-Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss synaptic transmission. Most communication between neurons occurs at a specialized structure called a synapse. A synapse is an area where two neurons come close enough to one another that they are able to pass chemical signals from one cell to another. The neurons are not actually connected, but are separated by a microscopically small space called the synaptic cleft. The cleft is less than 40 nanometers wide. By comparison, a human hair is about 75,000 nanometers wide. The neuron where the signal is initiated is called the presynaptic neuron, while the neuron that receives the signal is called the postsynaptic neuron. In the presynaptic neuron, there are chemical signals called neurotransmitters that are packaged into small sacs called vesicles. Each vesicle can contain thousands of neurotransmitter molecules. When the presynaptic neuron is excited by an electrical signal called an action potential, this causes the vesicles to fuse with the presynaptic membrane and release their contents into the synaptic cleft. Once they are in the synaptic cleft, neurotransmitters interact with receptors on the postsynaptic membrane. They bind to these receptors and can cause an action to occur in the postsynaptic cell as a result. This action may involve increasing the likelihood that the postsynaptic cell will become activated and fire an action potential, or decreasing it. Eventually, the neurotransmitter molecules must be cleared from the synaptic cleft. Some of them will simply drift away in a process called diffusion. In some cases, the neurotransmitter is taken back up into the presynaptic neuron in a process called reuptake. Once back inside the presynaptic neuron, the neurotransmitter can be recycled and reused. In other cases, enzymes break down the neurotransmitter within the synaptic cleft. Then the component parts of the neurotransmitter can be sent back into the presynaptic neuron to make more neurotransmitter.
In the last module, we took first steps towards understanding the electrical properties of individual neurons. We learned how electrical forces and diffusion give rise to membrane potentials, and we learned how cells can generate and propagate signals called action potentials, or spikes, along the membrane. Understanding the properties of the neuronal membrane is essential, but understanding just these properties isn't sufficient to give us insight into collective behavior of the billions of connected neurons in our brains. Luckily for us, we can approach neuroscience at many different scales and levels of analysis, and we don't have to confront the full complexity of everything all at once. That's what we'll be exploring throughout the rest of this course as we slowly go from our understanding of single molecules, such as ion channels, to the electrical behavior of neurons, to their collective behavior in small circuits, and finally onto how they become organized in large functional regions of the brain. Let's start simple though. Since we've examined one neuron, a logical next question is, how do two neurons connect with one another? We'll first examine some basic cellular anatomy of neurons. So far, we haven't made too much of the fact that the majority of neurons are polarized cells. That is, they have one portion of the cell for receiving inputs and another portion for sending outputs. The parts of the cells that are specialized for receiving inputs from other cells are called dendrites. The word dendrite comes from the Greek word dendron, meaning tree, and as you can see, the dendrites have a branching, tree-like structure. A signal received by a dendrite is passed to the cell body. If there is sufficient depolarization of the cell body membrane to initiate an action potential, then an action potential is sent down the axon. The axon then carries the propagating action potential to another neuron. So what actually happens at the boundary between two neurons, between the axon of one neuron and the dendrite of another? This interface is called a synapse. There are two general types of synapses: electrical synapses and chemical synapses. Electrical synapses are less common in our own nervous systems, but they're simpler to think about, so let's start with them. Electrical synapses are basically pores between two cells that allow ions to pass through. They allow the passage of the electrical signal through to a neighboring cell without much fuss. It's not so different than just combining two cells into one larger cell. There are lots of reasons that nature might need synapses like this from time to time. They're fast, and they allow cells to couple together with a high degree of synchronicity. But most neurons are connected together by a much more complicated structure called a chemical synapse. In a chemical synapse, rather than simply passing along an electrical signal from one cell to another, the action potential travels to the end of the axon and causes a chemical to be released into a very small space between the two neurons called the synaptic cleft. This chemical is taken up by the downstream neuron on the other side of the cleft. This chemical signal can cause the downstream neuron to depolarize its membrane, converting the chemical signal back into an electrical one. Or it can have other effects on the cell. This chemical step is slower than transmission across an electrical synapse, but it opens up an enormously diverse repertoire of different and more complex kinds of signaling. And synaptic function plays a critical role in computations performed by neurons. We'll spend the rest of this unit exploring the inner workings of chemical synapses, on our way to beginning to look at how networks of interconnected neurons give rise to behavior. We'll also look at the role of defective synaptic physiology in neurological and psychiatric disorders, and we'll see how synapses can be targeted by various psychoactive drugs and poisons. Finally, we'll wrap up by looking at how synapses can change over time in response to external stimuli, playing a foundational role in how we learn and
Hi, it's Paul Anderson, and in this video I'm going to talk about the synapse, which is a connection between adjacent neurons. That's a pretty strict definition. Remember, neurons can also be connected to effector cells in our body. But if we look at this neuron here and this neuron, the information has to get from this axon to this cell body or to this dendrite, and then the same thing down here. And we use synapses to do that. We're mostly going to be talking about chemical synapses in this video. In a chemical synapse, you have an action potential move down an axon. It triggers the release of neurotransmitters into this gap or this cleft right here. They're going to dock with chemically gated channels on the other side, and that can lead to an action potential on the other side. Some of the early work done with neurotransmitters was done by Otto Loewy. He had this dream of a wonderful experiment in the middle of the night dealing with the heart of a frog. He wrote it down, but when he woke up in the morning, he couldn't read his handwriting. Thankfully, he had that same dream again, and this is how the experiment that led to a Nobel Prize went. He had the heart of a frog that was beating, and then he would stimulate the vagus nerve on the surface of the heart, and what that does is slow the beating of the heart. So we're sending a message to the heart. He then took another heart from a different frog, but he removed the vagus nerve, so there's no neuron there. He got it beating, but he moved some of the liquid from the stimulated heart to the non-stimulated heart, and it slowed down as well. What did that mean? There was something in the liquid. He discovered these neurotransmitters, and he discovered a chemical synapse. In a chemical synapse, a action potential moves down a nerve, down a neuron. It triggers the release of neurotransmitters into the gap that are going to dock with receptors on the other side. Now, that's only one type of synapse. We also have electrical synapses. In an electrical synapse, an action potential is going to move down. And again, that's triggered by these voltage-gated channels. Once it gets to that synapse, it's going to open up more voltage-gated channels, and the message is going to go. An electrical synapse, if you have an action potential in the neuron before the synapse, you're going to have an action potential in the one after. What's great about electrical synapses is that they're very, very fast. What's bad about them is that there's no control. So let's talk about chemical synapses to identify the different parts of it. So this would be where the information is coming in. So on this neuron, and this would be the cell body of the adjacent neuron. So we have a presynaptic side and a postsynaptic neuron. And then this little gap in the middle is going to be synaptic cleft. Now, there are structures that hold this in place. It's not just floating there. On the inside of that terminal bud, we have synaptic vesicles, and those are filled with neurotransmitters. Those are different chemicals, chemical messages that can be sent across that synaptic cleft. There's some other anatomy that we should cover. There are going to be voltage-gated channels on the terminal bud. Those are going to be calcium ion channels. We also have docking proteins for vesicles on the presynaptic side, and then we're going to have receptors on the postsynaptic side. And so if we remove all of those terms, let's see what happens. So first thing that happens is we have an action potential that's moving down this neuron here. If you don't understand what an action potential is, I'll put a link to a video that I made on those. So the action potential comes down, and what that's going to do is the depolarization of that neuron is going to open up these calcium voltage-gated channels. Now, the electrochemical gradient for those calcium ions is into the neuron itself, and so they're going to move in, and they'll dock with chemicals on the surface of those vesicles. They're going to activate proteins on those surfaces, and what allows those to do is to connect with other docking proteins on the surface of the presynaptic side, and allows those vesicle membranes to merge. They then dump their neurotransmitters out into the gap where they can dock with these chemically gated channels on the other side. So they're sending a message through those neurotransmitters. Now the neurotransmitters will quickly then break apart. Some are broken down, some are recycled back into those synaptic vesicles. But let's see what's going on. So there are a number of different channels in every neuron. There are going to be leak channels for sodium and potassium. What those do is establish this resting potential. We also have voltage-gated channels. Those are important in the transmission of that action potential. But here we're talking about receptors. These are chemically gated channels. So when the neurotransmitters gap with it, they can open it up. What does that do? Well, it can move us towards or away from an action potential. So an action potential is when we hit this threshold of negative 55 millivolts. That's determined by these voltage-gated channels. But what can happen in an excitatory neurotransmitter, and a neurotransmitter that's pushing us towards the threshold, is that it is increasing the voltage. It's moving it closer to that threshold. What's a quick way to do that? If we have a receptor on the postsynaptic side that allows the flow of sodium into the neuron. That sodium is quickly going to depolarize the neuron and move us closer to the action potential. Now, some of those neurotransmitters may be inhibitory. What they're doing is they're allowing the flow of ions, but these are ions that are moving us away from that action potential. What's a quick way to do that? We could release a bunch of potassium or allow chloride to come into the cell itself. But if you think about it, a neuron is then receiving information from all the neurons that are connected to it that are telling it to either fire an action potential or not. What are they really doing? They're sending either excitatory, those are depolarizing messages, the influx of sodium that is starting to trigger an action potential, or inhibitory. Those are ones that are allowing potassium out or chloride in, and they're stopping that message from being sent. And so we really have an analog side to a neuron and a digital side. The summation of all of those signals, all those neurotransmitters, either tell that action potential to fire or not, and it's going to move down that axon. Now remember, when we get to the end, there are going to be terminal buds here. Again, calcium gates are going to open up, and we're going to have those new neurotransmitters that are sent across. Now, when you remember things, so if you can remember things early from this video, a lot of that has to do with connections in neural pathways in your brain. And what we're starting to find is that when you really remember something, that as we fire action potentials, as we have more of these synapses being activated, what we have is long-term potentiation. And so if we look at what's going on here, if we keep firing this neuron, we've done this in the lab, we keep firing and releasing neurotransmitters. On the postsynaptic side, the cell will actually develop and build more of these receptors, so it's more susceptible to that. And so we're building pathways of memory over time, and that's really what memory is. It's a set of neural pathways that are firing inside your brain. So that's the chemical synapse, and if you can remember a lot of the parts on this pre- and postsynaptic side, you're well on your way to improving your memory, and I hope that was helpful.
So as we have seen in the videos, so these diagram shows the neurons, dendrites, axons, which start from the main body of the neuron, and there are various branches, and the electrical signal, as you see here, it travels through this axon, right? Then here this is this synapse, and what was explained in this video, this is the synapse, and it is this synapse, it is shown in the amplified version of this. So it shows the gap between this two dendrites of the neighboring neuron. It is the synapse, and these are receptors here, and that is how the neurotransmitters are released, and the electrical signal it propagates. Now these neurons are further classified into categories like sensory neurons and motor neurons. So the sensory neurons carry information to the brain. So for example, through skin or eyes or other various types of sensory organs, the information is sensed and then it is transmitted to the brain. So that is done through sensory neurons. Now the other function of these neurons is to carry the signal from brain and convey to the muscles for taking some action. So those type of neurons are called as motor neurons. So these motor neurons carry information from the brain to the muscles, and there the third type, it is the interneurons, convey the signals around the nervous system. So these are different types of neurons: sensory neurons, motor neurons, and interneurons. So this you can see in this diagram, the sensory neuron, motor neuron, and interneous neuron. Neurons can be classified by their shape. So based on the shape also, the neurons can be classified as multipolar neurons, bipolar neurons, or monopolar neurons. So in case of multipolar neurons have many dendrites, whereas bipolar neurons have one dendrite and one axon. Whereas monopolar neurons have only one projection from the soma, which branches to the axon and the dendrite. So you can see the actual diagram of this, then you will understand what do you mean by multipolar, bipolar, and monopolar type of neurons. So the first one you see here, this is multipolar neuron, have multiple dendrites, what you see. Then bipolar neurons are composed of a single dendrite on one end and the single axon on the other end. So this side it is a single axon and one side there is a single dendrite. Then monopolar neurons have only single extension that leaves the soma and branches in two directions. So these are different types of neurons. Then earlier we also discussed about the glial cells. Apart from neurons, in our nervous system, there are the other type of cells called as glial cells. So the main function of these glial cells is speeding up the neuronal signaling and regulating extracellular chemicals, and also they enable neurons to modify their connections. So this is the structure of glial cells. So we'll stop at this point, and next time we'll discuss more about this synaptic transmission. What exactly is the synaptic transmission means how actually the electrical signal it is transmitted from one point to the other point, and that also briefly we have discussed in one of the video. You have seen in one of the video, it releases neurotransmitters at the synapse, right? Then there are receptors, postsynaptic potentials, and so on. So at this point we'll stop. Next time we'll continue from this point.